Hi everyone, this is Kate. If you don't recognize me from my intro video, hi, it's nice to meet you. Today I'm gonna go over the leucine zipper with everybody. I'm gonna start by going over some principles that are involved in holding the two alpha helices together, some possible residue that could replace leucine, um, and talk about how there could be possible residues that could destabilize the zipper, and also go over some stuff that kind of resembles lysine and glutamate. Alright, so let's go over the structure of what an alpha helix is. So an alpha helix is a tightly wound coil that is bonded by hydrogen bonds. That's how it makes this coiled shape. And then the leucine zipper is actually two of these alpha helices bonded together in the middle. They have side chains. You can't see them here, but they're sticking out like little sticks. And they're interacting with one another, and then they form this tightly wound coiled coil. Okay, so just to talk about the stability of the leucine chain. So the leucine chain has a leucine amino acid around the peptide chain, and it's at every seventh amino acid. So that's kind of, you can see it in the red here. It doesn't look like it's every seven, but it's every seven. And these interact with one another in the middle. They actually form hydrophobic bonds right in the center. And these hydrophobic bonds are, we know, water-fearing, so the aqueous solution that is around it is repelled away from it. So I kind of think of it as forming a force field around the zipper so the aqueous solution can't come inside and break apart those bonds. So another part, something that contributes to the stability of the leucine chain, is lysine and glutamate. So these two are pretty important because they form electrostatic bonds in the middle of the two alpha helices. So these form these electrostatic bonds, which is positive, negative, acidic, or basic, and they are opposites, and opposites attract, and they form these bonds in the middle, and that essentially creates kind of a double stability thing going on with the leucine zipper. So it's super stable. So if I were to replace the leucine and the leucine zipper with anything, I would probably choose isoleucine because it is also hydrophobic, which is a super important part of the leucine zipper, but it's also a lost my train of thought. Oh, isomer of leucine, which is pretty important as well. It's also the same mass, and I think it would go really well instead. So another thing that could possibly destabilize the zipper, something that we probably don't want involved, is serine, because it is hydrophilic, and hydrophilic is water loving, so it would kind of invite the aqueous solution from the outside to come in, and it's basically just asking for it to be destabilized. Okay, so two things that are pretty similar to lysine and glutamate. So these two we know, lysine and glutamate, we know are positive and negative, hydrostatic, and form these bonds to create a nice and stable environment. So we want to maintain this. So we have histidine and aspartate, that's what I chose. So this also forms that electrostatic bond in the middle that will make a nice and stable bond. And something else that's also important to note is that they are also acidic and basic, which also create this bond. So it's important to know that histidine is basic and aspartate is acid. So these will essentially attract each other and they will form another bond, therefore making it even stronger as well. Alright, so that is the leucine zipper and what makes it nice and strong and stable, and some potential things that we could use instead of leucine and some things that we could use instead of lysine or glutamate.